Morning, everyone. Thanks for coming to the network biology track. I'm just going to do a brief intro before we start with our first talk. My name's Alex Pico. I had the pleasure of chairing the NetBio Cozy this year. Together with our organizing committee and dozens of reviewers, many of you are probably here in the audience, we selected from over 100 submissions. We selected 2 proceedings papers to be published and presented today. 13 abstracts were selected for oral presentation. 9 of them will be in a longer format, and four of them in a shorter format. And then we have over 60 accepted posters. You'll find them presented this evening at 6 p.m. With people standing next to them. And they're all prefixed with the letter M, so they're easy to find. They're on the left-hand side. And of course, in addition to those, we have 2 invited keynotes for today. So if you do plan on skipping around, though we have the most comfortable chairs here, so you really shouldn't be skipping around. Be sure you come back for our keynotes. The first one will be Christian von Meering at noon, right before lunch. And then Laura Furlong is going to finish out our session at 5:20 with her keynote. Like I mentioned, it's a team effort organizing this meeting each year as part of the NetBio Cozy. These are the other folks who helped put it together today's program. And each year we recruit new members. So if you think you might be interested in providing some fresh ideas and helping put together what we do next year for NetBio, come up to any of the session chairs that you'll see today in person or email us later. All right, without further ado, our first presentation will be by Torsten Gross from Humboldt University here in Germany, or next door in Germany. He's one of our 2 proceedings presentations, and he'll be talking about robust network inference using response logic. Please.
Yesterday I thought they were changing so that there's actually mirrored the 2 screens.
Right. Let's just close that one. Close that one. That's the only one open.
Do you know how to...
Is it the PDF?
Yes.
You need to gather the window and to mirror system.
Of course, in rehearsal, it was all smooth. And now I've got to look there. Boy.
Yeah.
Arrangement. Gather and mirror. Great. One more try.
Oh, it looks promising.
Voila.
All right, thank you. So, good morning, everyone. I want to talk about network inference. So, network inference became big with the onset of the omics age, because then we were able to quantify various types of components of cells, but what was missing was a link and a way of gaining some mechanistical insights in how the behavior of certain components relate to the behavior of some other components. And so over the years, various types of methods and algorithms were proposed, and this, nevertheless, still network inference stays a hard problem. And that is simply because it is extremely context dependent. Like, your choice of method will very much depend on the context that you're in. And so this this actually makes network inference hard, and I hope that this project can help to alleviate a few of these problems. So my name is Torsten Gross. and I work in the Blütgen lab and in Berlin. And one of the main areas of interest of our research group is to study signaling pathways in a cancer context. So we're interested in trying to decipher what are the topological changes in our effective network models that could relate and explain certain of the phenotypical insights that we see. And a typical way in which we do is to look at perturbation type of data. So this is data that describes how the activity of some of the kinases in our pathways changes upon perturbation of other kinases in it, for example, through certain inhibitors. And so I tried to pursue this goal and went to the literature and tried various different methods. And what I realized is very soon, like with every method that I tried, I got a different result. And that makes a lot of sense because every method has different assumptions. But what the real problem is that it's extremely difficult to validate or evaluate the validity of these different assumptions. And so I asked, isn't there a way in which I can try to come up with a scheme that makes network inference or allows you to have a sort of intuitive understanding that tells you, okay, so here's my data, and this is the result that I get from it, and how will this result change when I change certain of my data points? Something that makes assumptions that are so mild that they're this scheme will be applicable in a broad range of topics. And also I want to have something that is adaptive. And by this I mean something that allows me to incorporate my domain knowledge. Because remember, these signaling pathways that we study have been researched for decades. And so I just don't want to discard all of this knowledge, but I actually want to use it. And I had an idea. And I call it the response logic. And it's blatantly simple. So let me explain. So for this talk, I want to consider directed networks, such as this one here. And the idea is simply that I say, if I have a perturbation at one node, it will get propagated downstream its edges so that every node that is connected to the perturbed node, and there's a path between them, will actually also feel the response. So, and this sort of reachability relation is also called the transitive closure. And so let's go into an example. Let's suppose I were to perturb node 3 here. Then you see that these red-colored nodes would actually feel the perturbation, and the others don't because they're not connected via a path. And so this is just a matrix representation of this idea. And now I assume that I can measure such type of transitive closure, but really what I measure is not exactly this, but a sort of a blurry version, which I want to call the response pattern. And so this can have missing data. For example, here I wasn't able to perturb node 2. And there can also be misclassifications, so disagreements between my transitive closure and the response pattern. So for example, here I see a shade of blue where the according square here is red. And now I want to see, can I take this type of data and assume that my response logic holds and also formulate all kinds of prior knowledge? And from this information, I want to find a transitive closure that is the most close to the response pattern and also conforms to these various types of information. And then I want to see what are all the possible networks that could have given rise to such a transitive closure. And note that this could actually be a gigantic set. So if I only do very few perturbations, you can imagine that my problem is very underdetermined. And so this will be very large. And I actually see this as an advantage of my method because then you're not just heuristically picking out a single one here, but you're actually, you get a feeling for which links in your network are actually identifiable given the data that you provide. And so if this is very big, then it might be not very handy to actually consider them individually, but I might resort to some sort of a union where you have edges, like here the black one that tells you there is an edge in all of these networks up here, or somewhere there's not, and somewhere there's a bit of ambiguity. But so this raises a sort of a technical issue here, because if you were to simply parse through all possible network structures, you would have to go through such an astronomical number here, and that's simply not feasible even for small networks. And so the solution that I found was to use logic programming. And so if you haven't heard about it, I want to give you just a 1-minute hint of what it can do for you. And so the specific language, logic programming language that I use is called answer set programming. And if you Google it, you find that it offers you a simple and powerful modeling language to solve combinatorial problems. And so the idea is that this is no longer an imperative language like what we usually do in Python, R, or C, or whatever you use, but rather what you do is a declarative language which allows you to describe your problem in a formal way. And so for my response logic, this can look like something like this. So this is really just to give you a flavor of what it does. The actual syntax doesn't matter so much right now. But so here I simply say, okay, so consider n nodes and consider that there could or could not be an edge between any of these pairs of nodes. From a certain combination of edges, define a transitive closure, and then simply compare the transitive closure to my response data. And so if there is agreement, I want to consider this type of a network, and otherwise I want to discard it. And so then you can take such a description of your problem and pipe it into a dedicated solver. And these solvers have been developed over the years, and they allow you to highly efficiently parse down your search tree. And this allows you, so in my case, this allows me to infer networks of up to a few hundred nodes in reasonable runtime. And a crucial advantage of such an approach is that it allows you to formulate all that prior knowledge in a very coherent way. And we're going to see a few examples of this in a minute. And so first I've asked, does it actually work? And so I resorted to something that I consider sort of a standard in benchmarking network inference methods, which is the DREAM challenges, so in particular DREAM 3 and 4. And to give you a brief idea of what it is, so the organizers of the challenge, some of whom I actually think might be in the audience right now, designed a few artificial synthetic networks that represented gene regulatory networks. From that, they did simulations of perturbation experiments and passed on the steady states to the community, who would then use reverse engineering methods to make network predictions, which could then get compared to the gold standard and scored in a standardized way. And so this allows me now to do the same thing and use this data to compare myself to the methods that were used at the time. And you see that the response logic performs relatively well. So except for these small cases, but in defense of response logic, I should probably add that the winning methods here made heavily use of time course data that I didn't choose to use in this approach. Okay, so now I felt confidence that this might actually be helpful to describe real data that was generated by my collaborators. And so they looked at signaling pathways in a specific colon cancer cell line. And so this is parts of the MAP kinase and PI3 kinase pathway. And they perturbed it at these 4 positions here. And you see the resulting response pattern right here. And now I ask, so does my response logic actually work for this type of data? And so I wanted to see if it can re-establish the known literature knowledge, which I've depicted here. And so I just computed standard scores for binary classifiers, which are the precision recall area under the curve and the ROC area under the curve. And so if you would just naively plug in the data, you go to this first line and you see that I'm just performing barely better than what a random classifier would do. And this actually makes a lot of sense because, see, this matrix here is very long. So we have 10 or 11 nodes to only 4 perturbations. So the problem is very underdetermined. And so I wanted to see, so can I put in some additional knowledge that is sort of generic, something that we know about these signaling pathways, but not specific to this very system here. And so I tried to say, so I want to exclude receptor target links, because the whole idea of the signaling pathway is that the signal gets sort of mediated, sort of transmitted through some intermediate steps. It was observed that signaling pathways are typically sparse, so I include a sort of a parsimony constraint here. I want my receptors to actually have an output. And so when I do this, I end up with these scores, and my set of solution reduces to 66 networks. And now I can compute amongst these 66 what is the fraction for each link of whether it's present or not. And so if I use this as a score in my binary classifiers, I come to these scores here in the end, which are highly far away from what a random classifier would do, which gave me confidence that I can go ahead and actually try to learn something new on this data set. And so what we tried to explain was some qualitative differences that we saw when we looked at mutants, so isogenic cell lines that were mutant in the PI3 kinase. And so PI3 kinase is closely related to AKT over here. And so this time in this modeling approach, I didn't try to resort or to reestablish a known literature network, but rather I wanted to really see what are the specific differences between my cell lines. And so the idea was that I actually constrain my literature knowledge, except at all the places where I could expect some variance due to these different mutations. And so I talked to my colleagues who are experts in the signaling, and we found out, so we just decided that AKT, due to the mutation in PI3 kinase, so all edges going in and out of AKT should be something that the algorithm is free to infer, as well as all the ones that go into EGFR, because this we have seen many times that EGFR is actually, the feedbacks into EGFR is something that you see or see not very much depending on a certain context of the cancer. And so I let the program run with these constraints, and this is the results you get. And just to highlight the two key features that you see in here is that the mutants actually break the link between the EGFR receptor and these PI3 kinase pathway members on the left, and that for this green mutant cell line, we have actually a feedback loop here. And so these were the hypotheses that we made, and then we asked, so can we actually confirm this? Do we see some phenotypical changes that we can explain with these? And so we measured various drug response curves, and I want to show you two of them. So one is that, so we measure growth of these cells after perturbation with MEK inhibitor. So MEK inhibitor will eventually reduce the activity of ERK here. And so what you see is actually that this green mutant here is the most resistant, and that makes a lot of sense because of this feedback that we've seen here. That is something that you see a feedback gives you a certain type of robustness, something that which has been reported in the literature many times. And then also looking here at what happens in the EGFR receptor inhibition, you would expect, since the mutants break the link to the PI3 kinase pathway, that they would actually be more resistant. And this is what you find, but only for this orange cell line here. The green one, however, we hypothesized that due to this feedback here, our MEK kinase pathway activity is already so low that if we further reduce the activity of EGFR, we actually bring it to subcritical levels. Okay, and so with this, I want to just wrap up and remind you that I hope that the response logic is a way, or is an intuitive and broadly applicable and adaptive way of doing network inference, that it solves a hard combinatorial search problem with the help of answer set programming. It outperforms some of the competing methods in dream challenges, and it helps us to explain mutant-specific sensitivities to targeted drugs. And so we think that actually it might be very helpful to use this method in a new type of data that comes up now, which is that of single-cell perturbation data. So methods like perturb-seq or crop-seq now give you this new class of data, and we're excited to use it. And so if you want to also try it out and see also different types of data sets where I applied it that I wasn't able to show here, you can just resort to this repository. So thank you.